Hello everyone, welcome back to my channel Biomix Hub. In this video, we are going to discuss about RNA interference, and it is a topic of molecular biology. It comes under MSc and other different competitive exam and also in CSIR Life Science Net. In this video, we will discuss about miRNA, and in the next video, we will discuss about siRNA and solve some of the questions which are asked in CSIR Life Science Net. So let us start this video now. So, RNA interference. RNA interference is also known as RNAi. Now, what is the function? It is interfering with the process of RNA. So, RNA interference are of two types. They are the siRNA and the miRNA. In this video, we will discuss about miRNA, how this miRNA is formed and what are the processes taking place for silencing of the mRNA. So, first of all, we will discuss how this miRNA is formed. In the nucleus, what we have the DNA. From the DNA, the mRNA is produced through the process of transcription. mRNA is produced and this mRNA is translated into protein. There are some protein through the process of translation. Protein is produced. There are certain proteins which are harmful, or there are some proteins which we don't need. We have to eliminate that protein. For that process, we need miRNA or siRNA, by which the proteins or the genes which are not needed or the harmful genes can be silenced. Now we will see how the miRNA is produced. The miRNA can be both endonuclease as well as exonuclease. That means we can insert miRNA from outside into the shell. Now see, in the nucleus we are having the DNA, and from the DNA mRNA is produced. mRNA is produced. This mRNA can be converted into miRNA. How it will be converted? What happens? This mRNA which is produced from the DNA sometimes have the complementary region. Here it will have its complementary region. Then what is taking place here? These two complementary regions will bind with each other. So after the binding, they will form a loop-like structure, and these are the complementary regions which will bind with each other. This is a miRNA which is producing a loop-like structure. This is the structure of miRNA. Now we will see what are the processes taking place for the maturation of the miRNA and how the proteins or the mRNA are silenced through miRNA. We will see what are the processes taking place in the nucleus for the formation of pre-miRNA. What happens in the nucleus? When the DNA is present, mRNA is produced, and for the formation of this mRNA, we need RNA polymerase 2, and sometimes RNA polymerase 3 is also responsible. And the miRNA is produced when the mRNA is having the complementary region. That means here the DNA is giving the mRNA, and if this mRNA is having the complementary region, then it will form the miRNA by forming a loop, and this miRNA is known as pre-miRNA, pre-miRNA, because this is not matured now. Now what is miRNA? Is it coding or non-coding? When we talk about miRNA, it is a non-coding RNA. What do you mean by a non-coding RNA? That means it is not responsible for the formation of any kind of proteins. And what is its length? The length of miRNA is 20 to 25 mR. Okay, now we are done with this. Now, if the miRNA is produced where it will bind for the degradation of mRNA or silencing the mRNA, when this miRNA is produced, it will bind to the 3' UTR region of mRNA. Okay. Now let us see how the maturation process of the pre-miRNA will take place. The first process takes place in the nucleus. See, here we are having the pre-miRNA, and in the pre-miRNA there are some extended regions. These are the extended region. First of all, this region should be cleaved, and for cleaving this region there is an enzyme known as DROSA, and this DROSA is a RNase 3 endonuclease, and it is double stranded RNA specific. Okay, it is a RNase 3 endonuclease, means it is a kind of RNase 3 enzyme, and it is endonuclease, that means it will cleave from the middle of the region. Here from the middle it will cleave. Okay, along with DROSA there is another protein known as PASHA working together in case of Drosophila. If here DROSA is present, if here DROSA will be present, here PASHA will be present for the cleavage to be done. And if it is mammal, along with DROSA, DGCR8 will be present in case of mammals for the formation of pre-miRNA. Here first there was pre-miRNA, and after the processing with DROSA or PASHA in case of Drosophila, or DROSA or DGCR8 in case of mammals, there will be formation of pre-miRNA. After the formation of this pre-miRNA, it will be exported out from the nucleus. And when we talk about the DROSA and PASHA, when it will combine and work, it is known as microprocessor. Through the microprocessor, the pre-miRNA will be converted into pre-miRNA. Here the microprocessor is DROSA, the function of DROSA and PASHA which is performed. These two are known as microprocessor. It can be DROSA and PASHA in case of Drosophila, and in case of mammals it can be DGCR8. Okay. Now what happens? It needs to be exported from the nucleus. So for the export to be done, it needs a channel, and it is the XPO5 or the exportin 5. Through this it will be exported from the nucleus. Now after the export from the nucleus, what it will be done? This is a pre-miRNA which is exported from the nucleus into the cytoplasm. Now in the cytoplasm there is another enzyme known as dicer. Now this dicer will cleave this loop and will form a overhang of 3' OH. Okay, this is a miRNA duplex will be formed here. The dicer will cleave this region and it will form 3' OH overhang and 5' phosphate. Okay, here also 3' OH overhang will be produced and 5' phosphate will be produced, and this is a double stranded miRNA duplex. Okay. Now when we talk about the silencing of the mRNA, how it will be silenced? Here we are having a duplex. Here a duplex is present. Now this duplex will be forming a single strand. One strand will be cleaved for the process to be done. Why this need to be cleaved? We will understand that. Here only one strand will be present and one will be degraded. Now we have to know which strand will be degraded and which strand will work. Now this miRNA should come and bind to the 3' UTR region of mRNA. Suppose this is a mRNA which is having the 3' UTR region. This is the 3' UTR region, and this mRNA, this is a mRNA, and here this mRNA should be silenced. So miRNA is responsible for the silencing of mRNA. So the miRNA should come and bind to the 3' UTR region of the mRNA for the silencing process to be done, due to which there will be no formation of the proteins which was harmful or the proteins which we don't need, which we want to silence. So which strand will come and bind here? The strand which is complementary to this region, to the 3' UTR region of the mRNA, will come and bind here. Suppose this is the strand which is complementary to the 3' UTR region, it will come and bind here. This strand will come and bind here. The other strand which is present over here, as it is complementary with this, it should be same as this region. This strand should be same as mRNA region. So the strand of the miRNA which is same with the mRNA region, it will be degraded, and which is complementary to the 3' UTR region of the mRNA, it will come and bind to the 3' UTR region of the mRNA and it will silence. Okay, due to which there will be no translation, there will be inhibition in the translation. So for the process to be done, we need another enzyme known as agonut. Okay, here we are having only one single stranded miRNA, which is complementary to the mRNA 3' UTR region. So this is a mature miRNA. Okay. So after the mature miRNA is formed, it will form a RIS complex. Okay, how is a RIS complex formed? It is formed by the protein agonut. Here there will be formation of RIS complex through another protein which is agonut. Agonut will come and bind here. It is also known as AGO. Okay, when the agonut and the RIS complex is formed, then it will bind to the 3' UTR region of the mRNA, due to which there is inhibition of the translation and the mRNA is decayed. Now we will see how it is inhibited. Here we are having this is the RIS complex along with miRNA. Okay, along with AGO. AGO is a protein which is responsible for chopping. If this miRNA which is present is perfectly matching with the 3' UTR region of the mRNA, if it is having total complement region with the 3' UTR region, then what will happen? It will be chopped up, and this chopping is done by the protein agonut. Okay. If the miRNA which is present, it is binding with some little portion and some portion are not binding with it, then what will happen? There will be no chopping, only it will be inhibited during the process of translation. We are clear with this? Again, let us discuss this. If the RIS complex along with the miRNA is totally complement with the 3' UTR region of the mRNA, then the process of chopping will be done with agonut. If only some portion of it is binding, say here in this region the total portion is complement with the mRNA, the miRNA and the mRNA is totally complement, here chopping is done. In this region only some portion of the miRNA and mRNA is complemented, here in this region it is not complemented. Here no chopping is done. Here there will be no translation, but chopping is not done here. Okay, we are done with this. So now we will discuss what is the difference between the plant and animal silencing. So in case of plant shell and animal shell, the silencing is done, but the only difference is with some of the proteins and the place where it is done. Okay, let us see the difference now. What happens in case of animals? This A is the animals and this is a case of plants. Let us see the difference. In this case, so the RNA polymerase 2, we are forming the pre-miRNA. Pre-miRNA is produced, and for this cleavage to be done, there we need two enzymes. They are the DROSHA and PASHA in case of Drosophila. Here see the enzyme DROSHA is present, here PASHA is present. Okay. So here the external part, this part are digested. What were the DROSHA and PASHA? There was a RNase 3 endonuclease, that means they will cut from the middle. Here the processing is done. Okay. After the processing is done, after this region is cut or it is processed, then this pre-miRNA, this pre-miRNA is converted into pre-miRNA. This pre-miRNA is now transported into the cytoplasm from the nucleus, and for the transport process to be done, it needs exportin 5. This is exportin 5. After it is transported into the cytoplasm, there is another enzyme known as dicer. Here it is, I am writing here, dicer. This dicer is responsible for cleaving this region. It is cleaving this loop-like structure, due to which it will form the miRNA duplex. And this miRNA duplex, one strand will be degraded and another strand which is complementary to the 3' UTR region of mRNA. Suppose this is a mRNA, it is having the 3' UTR region. Only one strand will be complementary with this 3' UTR region, and another strand will be same as this 3' UTR region. The strand which is similar with this 3' UTR region will be degraded, and the strand which is complementary to the 3' UTR region will be bonded here, due to which there will be degradation of this mRNA, or further processing will not be done. Okay. Now we will see what is the condition in case of a plant, what are the enzymes responsible for the process to be done. In case of a plant, this pre-miRNA is produced. For the processing of this pre-miRNA for the formation of pre-miRNA, in case of animals there was DROSHA and PASHA, but in case of plants there is DCL1, and its function is like DROSHA, and the function of PASHA is done by HYL1 in case of plants. These are the major difference between the plants and the animals. Here pre-miRNA is produced. After the pre-miRNA is produced, what was taking place in the animals? It was exported into the cytoplasm. But in case of plants, the next process is also done in the nucleus. That is, the formation of this duplex is done in the nucleus itself, and it is done by this DCL1. Okay. Now the DCL1 is acting as DROSHA as well as dicer, because inside the nucleus, see here DROSHA was working for the conversion of pre-miRNA into pre-miRNA, and dicer was working for the formation of pre-miRNA into miRNA. So the both work is done by the work of DROSHA as well as dicer is done by DCL1 in case of plants. See here we are having pre-miRNA over here. The process from pre-miRNA to pre-miRNA is done by DCL1, and the formation of pre-miRNA into miRNA duplex is also done by DCL1. Now after the formation of this duplex, it is exported into the cytoplasm from the nucleus. So for the export process to be done in case of animals, exportin 5 was responsible, but in case of plants, it is HASTY which is responsible for the export process. See here, after the export, here methylation is done in case of plants, but in case of animals there was no methylation done. Okay, and the process of methylation is done by HEN1. And where is this methylation taking place? The methylation is taking place in the 3' OH region. See here, the methylation is taking place, and here the methylation is taking place. The rest all work are similar with the animal. Okay. How? The strand, here we are having the double strand, the strand which is complementary to the mRNA, the strand which is complementary to the mRNA is coming and binding in the region to the 3' UTR region, and the strand which is same to the 3' UTR region, it is degraded. Okay, by this there is silencing of the mRNA, or the work of mRNA for the processing of the protein is not done, by which we will not get the protein which was harmful, or which proteins we want to silent, we can silent in this manner. This is all about the processing of miRNA, and in the next video we will discuss about the process of siRNA, and we will discuss some of the questions of CSIR Life Science Net. And thank you for watching this video till the end. If you like this video, please do like, share and subscribe. Thank you. Bye bye. See you in the next video.